welcome all participants of the conference. And I would like to present my report to topic pharmacological flora of Azerbaijan. First of all, I want to present speed location of Vicia L species in Azerbaijan. And the genus Vicia L belongs to the legume family Fabaceae. Out of 150 species widely distributed in the northern hemisphere, 48 species grown in the Caucasus, 42 species in Azerbaijan, some of which are cultivated plants. Raw materials of species belonging to the genus Vicia L are used for the treatment and prevention of various diseases in folk and scientific medicine. Decoction of the flowers of the plant is used in folk medicine for allergies. It has been established that the acetone extract of Vicia faba L has antifungal activity. The activity of Vicia L raw materials against skin infections and against age spots of the face was studied, and also has some other effects. As introduction, the flora of Azerbaijan is distinguished by a rich variety of vegetation. Recently, this raw material has been used to fully meet the needs of medicine and health care. Research work is gradually being increased in the direction of sufficient study of the chemical composition and biological activity of plants and the determination of the raw material base. In the flora of the Republic of Azerbaijan, one of the widespread genera belonging to the legume family is Vicia L. And a number of studies have been carried out to study the chemical composition and biological activity of animal species. It was found that the amount of isoflavonoids such as genistein and formaldehyde is higher than in other species. For standardization of Vicia faba plant materials, microscopic and macroscopic properties, as well as physicochemical parameters were determined. Also, as a result of phytochemical studies, it was found that raw materials of some species belonging to this genus contain flavonoids, other flavonoids, hydroxycinnamic acids, vitamins, amino acids, and other important substances. The raw material resource of some species of the genus Vicia, common in Azerbaijan, have been studied, and accordingly, the areas of mass distribution have been determined. As a result of preliminary pharmacognostic studies, plant species Vicia faba L and Vicia sativa L, widespread in Azerbaijan, were recognized as promising as objects of study. As a result of phytochemical analysis, it was found that the surface parts of raw materials contain flavonoids and hydroxycinnamic acid, as known in the standardization of medicinal plant materials. In addition to the phytochemical composition and morphological features of raw materials, the study of characteristic diagnostic features in its anatomical structure is also of particular importance. Until now, no research has been carried out on the morphological and anatomical aspects of species belonging to the genus of Vicia L growing in Azerbaijan. The purpose of the work is to study the morpho-anatomical structure of raw materials of the Vicia Faber L and Vicia sativa L, which is widespread in our country and belongs to the genus Vicia. Now about methods. The plant Vicia Faber for research was harvested from the fossil zone of the Shamaho region of the Lesser Caucasus in June 2021 during the flowering period of the plant. To determine the anatomical and diagnostic features of the studied raw materials, the corresponding micro-preparation were prepared. Samples were prepared in 0.1 phosphate buffer, pH equals 7.4, in a solution consisting of 2.5% in glutaraldehyde, 2% paraformaldehyde, 4% sucrose, and 0.1% picric acid fixed and transformed to the electron microscopy. At first, the samples were washed in 0.1 phosphate buffer, pH equals 7.4 for 15 minutes each, 45 minutes total, 3 times after removal from fixative in the laboratory. At the post-fixation stage, sections prepared for microscopy were kept in a mixed solution of 1% of 0.1 phosphate buffer, 0.1 phosphate buffer, and 1.5 accelerated blood solution for 1.5 hour after post-faction using an unknown general method for light microscopic studies. After the fixation step, the samples were again washed 3 times for 15 minutes, 45 minutes in total, in 0.1 phosphate buffer, pH equals 7.4, and the sections prepared for section 3 were dehydrated. At this time, the samples are passed through 50% and then 70% and 96% ethanol solutions, respectively, for 15 minutes each time, 45 minutes in total. Then the micro preparations were washed with a solution containing 50% pure alcohol and 50% pure acetone, 3 times for 15 minutes, total 45 minutes, and fixed in pure acetone 3 times for 15 minutes, total 45 minutes. The next stage, fourth, was the preparation of blocks. For this solution of araldate upon was used, which consists of a mixture of 5 substances, 2.5 milli upon 812, 55 milli DDSA, which means the determinyl succinic aldehyde, anhydride, and 7 drops 0.3 milli dibutyl phthalate and 7 drops DMP30. DMP30 is dimethyl aminomethyl phenol epoxy, and their weight ratio varies depending on the number of samples taken. The stage of preparing a block with 75% pure acetone and 25% araldite upon 50% pure acetone, 50% araldite upon 25 pure acetone and 75% araldite upon. The composition of the solution is 100% araldite upon, which is divided into several parts. In turn, the micro preparation stands there for 1 hour. After the completion of the last stage, the prepared samples were placed in pre-selected and numbered ready-made form and placed in a thermostat. The parts prepared for cutting were kept in a thermostat at 37 Celsius for 1 day, 45 Celsius for 1 day, and 60 Celsius degree for 1 day, 3 days in total. Semazine sections were first prepared from ready-made blocks using a RESA AMUC7 ultra microtome. Sections prepared for analysis are subjected to double staining solution A, 0.5%, methylene blue, 0.5%, azure, 0.5%, borax, and solution B, 5% ethanol, 0.1%. The slide microscope and the shape of the necessary part were compared on a Canon D650 digital camera made Japan. After making pyramids from selected sections from the necessary part of half sections, viewing them under a light microscope, they were assembled into garlands prepared from silver and gold ultrazine sections, 50, 18 microns zinc, using the aforementioned ultra microtome. Image morphometric analysis was performed on electrograms taken in TAF format using a computer program. The TAN imaging platform developed by the German company Olympus Sophia Imaging Solid Ions and the article now about results. The article presents the results of studying the diagnostic feature of the morpho-anatomical structure of the Vicia L raw materials from the flora of Azerbaijan. Morpho-anatomical study of the medicinal plant material Vicia L revealed the following characteristic features of the structure. Stemzine thinly glabrous, it's a usually folder, curved and 35-50 centimeter long. The ovate, the leaves are composite of small, deeply divided ovate lobed parts. Leaves inverted ovate wedge shaped or inverted triangular, mangine, and especially fragile. It consists of 5 flowers on a thin pedicel and is shorter than petiole. The flowers just now. First of all, it's microscopy study of leaves of Vicia L. Now here results about roots. As a result of the conducted microscopic studies, the main diagnostic features of the anatomical structure of the roots, leaves, stems, and flowers of the Vicia L raw material were determined. Here results. Yes. Here results of fruits, microscopy results of fruits, and results for flowers. The flowers group is very fragile. It consists of 5 flowers on a thin pedicel and is shorter than the petiole. The flowers are small, 4-5 milli long as the calyx or longer. The calyx is usually half the length of the flower or shorter. Flowers are purple. All spheres are black. In usual, the spheres are separated from each other by the thin slit brown ovate triangular convex with black dots on them. Blooms in April, July. It's Vicia faba and Vicia teinifolia. And thanks for attention.